This is the first lecture on transcription. This will cover the process of transcription in bacteria. Many of the processes are the same between bacteria and eukaryotes, but they tend to be more complex in eukaryotes. So we'll cover bacteria here first. Remember that this is where we're at in the process here at transcription. So this is the process of making a messenger RNA from DNA, which will then be transported to make protein. Now, you have to remember our structures of prokaryotes and eukaryotes. Remember, prokaryotes being the bacteria here on the left-hand side. They don't have a nucleus. They don't have membrane-bound organelles. So their DNA is here out in the cytoplasm. And because of that, the process of transcription takes place right out here in the cytoplasm, and the process of translation, which is shown here. Now, in contrast, our eukaryotes have a nucleus. Their DNA is contained in the nucleus, so in order to use that DNA to make RNA, that process takes place inside the nucleus. And that's going to have some different effects depending on how that process happens and how it's used in association with translation as well. So in bacteria, from now on we're talking about bacteria in this lecture, the processes of transcription and translation can actually be coupled together. Because this all takes place in the cytoplasm, these two processes can be happening at the exact same time. So here you see the DNA. This little purple guy is the RNA polymerase. It's going along making the RNA, which is this red strand. As it's making that RNA, the ribosome, which is this green guy right here, it can also go ahead and attach and start making the protein right away. So these processes can occur at the same time, and they can be very closely coupled together to make the protein as quickly as possible. And that only happens in bacteria when you have none of these processes separated by membranes. And you can see this in actual real life up here in this electron micrograph. We got the DNA here, and the little dots on the DNA are the RNA polymerase. These big dots are the ribosomes that are starting to make the proteins. So here's our overall outline, and we're going to go through each of these processes separately, but just the overall idea. Here's our DNA, here's our RNA polymerase. Take a look at the polymerase. Looks very similar to the shape of DNA polymerase. Okay, it's got that hand kind of shape. It's going to bind. It's going to start making RNA and eventually terminate. So the same kind of steps that we've already talked about for replication. It's got to initiate. It's got to elongate. So this would be our elongation, and then has to terminate down here. Okay. Our bacterial promoter. Promoter is a sequence at the beginning of where it's going to start actually making RNA. So this is the sequence that the polymerase is actually going to recognize and say, hey, there's a gene here. I want to transcribe that gene. The gene it's going to transcribe is actually down here. So this is a sequence that we would say was upstream of that gene. And when we say upstream, we mean closer to the 5' end of the coding strand. If we say downstream, we say closer to the 3' end. It gives us kind of a directional sense of where these things are at in relation to each other. Now take a look for a second over here, the coding and template strands. So the polymerase is going to read the template strand. It's going to read from 3' to 5'. Okay, so this is the enzyme, like it's reading a book. It's reading from 3' to 5', and we call that the template strand. It's going to make the complement of that strand, and it will make it going from the 5' to 3' direction. So this is the same as DNA polymerase, where it can only add to that 3' end. These polymerases, same with either of them, they can only add to the 3' end, so they're always going to make the strands in that direction. So the other strand up here, it's not really used by the polymerase, but we call it the coding strand in that its sequence looks similar to the actual mRNA once we get it done. So remember, we're reading this part and we're making the complement in our RNA. So it looks like this sequence up here. So down here, if you see this RNA that was being made, it looks like this piece. But it was made by reading this and making the complement of it. Okay, so this promoter has two specific sequences. These are just in bacteria. One is called the negative 35 sequence. That means it's 35 nucleotides away from this transcriptional start site. So if we go 35 this direction, which is upstream, so we're going in a negative direction. We want to make our RNA in the positive direction this way. So it's 35 nucleotides away, and it has a very conserved sequence, so meaning it's similar in a lot of bacteria. That's TTGACA in the coding strand. And then we have the negative 10 sequence, so this is 10 nucleotides away from this transcription start site, and it is TA-rich, so TATAAT is the sequence for those. And those are going to be important for helping the polymerase find that sequence and bind to it so it can then start making the RNA. The RNA, the transcriptional start site, is always a purine and almost always an A. So it could be a G as well, but more often than not it's an A, which will start here. Okay, here's our polymerase down here in the corner. There are different parts to this polymerase. In bacteria, there's only one RNA polymerase to worry about, and it can synthesize all types of RNA. So our RNA is our RNAs that are found in the ribosome. mRNA is what we're mostly talking about here, and those are the messenger RNAs that are going to code for proteins. And then tRNA, transfer RNAs, are going to be used to help make those proteins once we get to translation. So this RNA polymerase can make all of those different types. The polymerase consists of subunits beta and beta prime. So that's here and here. So these kind of come together to form the hand. So if you imagine a hand that's gripping this other protein in the middle, here are the fingers and here's the thumb. It also has two alpha subunits, those are down here, and a sigma subunit, and it's called dissociable, meaning it can come off. So this one doesn't stay there the whole time, it's there for binding, and then it goes away. So the beta and the beta prime, so the big RNA polymerase part, this is the part that's going to synthesize the RNA. This part looks very similar to a DNA polymerase. Remember, they're both making nucleic acids, so that makes sense. Then we have the alpha subunits. These kind of form a scaffold for the beta subunits to come together to interact. So they help that binding, and they bind another sequence that's upstream of the two promoter sequences we just talked about, and they help enhance transcription to make it better, faster. And then we have the sigma subunit, and that's this one, this dissociable one here in the middle, and that's important for binding at the promoter. There are a lot of different types of sigma subunits, and those allow very specific regulation. So only certain types will be made at a given time, and those tell the polymerase these are the genes that we need to make right now. So those are important for helping regulate which genes are made at which times. All right, so here's how binding and initiation is going to happen. So our sigma subunit and our alpha subunits are going to be the ones that really help with the binding to the promoter. The sigma subunit is going to recognize the -10 and -35 sequences that we just talked about, and it's going to help that polymerase bind to those sequences. The alpha subunits are going to bind to upstream elements called enhancers, and they're going to help just stabilize this complex on the DNA. Now, so that's the binding part, and whenever you see this arrow here, that means there's a gene right there that's going to be transcribed. We're going to go this direction, and here's the gene. So the initiation part is kind of interesting. It does this weird sort of thing where the polymerase will start, the polymerase kind of stays in one spot and it pulls the DNA through it. it will start moving and then pull back, so that it ends up kind of scrunching these DNA sequences up. So the blue is the DNA, the two parts of the helix of the DNA, and it will make these short little RNA sequences, usually less than 9 nucleotides long. It will do this, make these sequences back up and scrunch this area up, and this sequence will be released. It'll do this a bunch of times before it actually makes a long enough sequence that it can then move on and elongate. And it's not completely clear why it does this, but it does this in eukaryotes also. So prokaryotes and eukaryotes both have this kind of weird initiation, start-stop problem, and they call it abortive initiation, abortive synthesis. So they start to make one and then they stop and it releases. They start to make it and then they stop and it releases. They haven't really found any sort of function for these little pieces of RNA. That's not to say they don't have one. We just don't know what it is, if there is one. But this is how it gets started. Eventually, it will make an RNA long enough that then it will keep going. And that's when the actual RNA gets synthesized. So when it makes a long enough RNA, so more than 10 nucleotides usually, it will eventually then release the polymerase to elongate. So polymerase is no longer stuck back and forth, back and forth. It releases it to elongate. At that point, the sigma factor leaves. It's done its job. It's bound. It's good to go. And now the beta subunits can make the RNA. Another important thing to note here is that the RNA polymerase does not need a helicase. It can unwind the helix itself in order to get in there to read the template strand and to make the RNA. Okay, so here's a close-up look at our RNA polymerase. Remember, so it has these two subunits, the beta and beta prime, and they are often called pinchers because they help hold on to the DNA. So they're holding that DNA in place so that they can read it. In this case, our template strand is up here. Okay, so it's kind of flipped from that other diagram we were looking at. And here's our coding strand down here. So the polymerase is unwinding unwinding here and rewinding the DNA itself as it's making the RNA. The RNA is exiting through another channel. You can't really see it here. It hasn't shown you it specifically, but it's exiting through a different channel than the DNA. That's to make sure it doesn't stay associated with the DNA. You don't want it to stay in there so that the DNA can't wind up and the RNA can't be released. The nucleotides, remember we're using NTPs here. So in DNA synthesis, we use DNTPs because it was the deoxy versions. Here, remember it's the ribonucleic acid versions. So they have an extra oxygen, so they're just the NTPs, not the deoxy version. Those are going to enter through a channel that's through the back of this slide. So it comes in through the back of the enzyme, and the enzyme can then use them to put them in place. Remember, for transcription, we're using A, G, C, and U. So uracil replaces thymine in transcription. Okay, and our termination. Termination can occur in two different ways, and it just depends on the certain gene, which one of these ways it occurs. So some genes have this GC-rich region, and that would be, it'll be here. It's coded for in the sequence, and it's self-complementary. So meaning once it's made, it complements itself close enough, and it's close enough together that it can form the hydrogen bonds in between the bases of the same strand here. And this forms what we call this hairpin right here. And that hairpin has secondary structure here, so it can help make some space in between the DNA and the RNA, and helps the RNA release from the DNA where it's been elongating, and help it to be released by itself. The other thing that helps with this is this series of U's. So this series of U's kind of signals to the polymerase, hey, we're done here. This is the end of the gene, and this is we're ready to be released now. So those two elements, the GC-rich region, which will form a hairpin, and the sequence of U's, then helps release the RNA, And when the RNA releases, the RNA polymerase comes off here. If I said DNA polymerase at any point in this slideshow, know that it's RNA polymerase. We're always talking about RNA polymerase. There might have been a few slips of the tongue there. Okay, so that's one way. The other way is through row-dependent release. Row is an RNA binding protein that can bind sequences here at the 3' end of the RNA, and it helps also put space in between the RNA and DNA and release the RNA from the DNA. So if it doesn't have the GC-rich and the U's, there will be sequences that row protein can bind and help with termination.